Okay, so welcome everyone to this EVOLTREE Online Webinar series, which is organized in the context of a topic on polygenic adaptation. And we've got some excellent scientists from around the world presenting their work. And today I'm very pleased to welcome Sam Yeaman from Calgary, Canada, who will be telling us a tale of two architectures. But before we introduce Sam, I would like to remind everyone that this session is being recorded and we are also streaming it to our audience on YouTube. We had a very high demand for this webinar and we are very, very happy to see that there is an interest in this kind of online seminar series. I would like to also thank my co-organizers for their support in organizing this, and it's Christian Rellstab, Santiago Gonzalez Martinez, and Stephen Cavers, who are also here with us today. So with this, I would like to hand over to Santiago, who will tell you just a few words about EVOLTREE and introduce Sam. So Santi, over to you.
Thank you so much, Eva. I'm going to be very brief. I just wanted to introduce the network for those people that is the first time that have come to know about EVOLTREE. So EVOLTREE is an European research network that has as ambition to link ecology, genetics, genomics, and evolution in the context of forestries and forest ecosystems to address. Oh, sorry. And sorry, I was muted, I think. So I start again. Thank you, Eva, for introducing me and for organizing this very nice webinar. So I just wanted to say a few words about EVOLTREE for those people that is the first time that know about our network. EVOLTREE is an European network that has as a goal to link different disciplines related to forestries and forest ecosystems, namely ecology, genetics, genomics, and evolution. And we are trying to address global issues that are affecting European forests as a whole, like climate change or the loss of diversity or the emergence of novel pests and diseases. So I would say that we are becoming quite an old network now. We have been around about a decade, and we have been evolving like a network to come where we are now. Now we are focusing in activities that are mostly related with the coordination of research across Europe, facilitating this scientific discussion like the one we are having today. We are trying to also identify future research needs for our community doing exercises like horizon scanning approaches and trying to target also different communities that are related to forest and in particular genomics of forestries. We have also a few initiatives that are directed to students and early career researchers, mostly providing training and a bit of financial support. And also we are keeping some databases on tree genetic diversity and genomics, and also other facilities that we put at the service of the community, of everybody that would like to work with us. So basically, this is EVOLTREE, and this is what we have been doing so far. And if you want to get more information, please don't hesitate to go to our webpage or like to contact directly any of us people involved in the network. So this is enough for EVOLTREE. Today we are here not to talk about EVOLTREE actually, but just to listen to Sam. We are very happy that he accepted our invitation. Professor Sam Yeaman from the University of Calgary in Canada is a very well-known researcher for us, for the forestry community, but also outside it, as he has produced very remarkable theoretical studies on genetic architecture and local adaptation in different kind of organisms. So many thanks for accepting our invitation. It's really great that you are here. And I would say just a very, most of you will know already his work, but he has done contributions, very important contributions on how complex traits evolve, and how to develop and refine the genomic methods that are used to study them. And I would say that I did already mention some theoretical work, but what is very amazing of Sam's work is that also this is then applied in multiple with multiple approaches, also involving experimental studies and comparative genomics, computational biology. So we are having a great opportunity today to have a really good insight into a series of interesting topics and have a good discussion. So this is all I have to say today. So I will just pass the word to Sam. He will be talking today about his main topic, that's it, genetic architecture, local adaptation, and the migration selection balance. And Sam, the floor is yours. Thank you so much.
Thank you so much, Santiago, for the introduction. That's very generous of you, and thank you very much to the organizers of this for the invitation. It's really a pleasure to have a chance to speak with everybody. I think this is a silver lining in COVID, that we can actually meet all around the world all at once and chat about science. So I hope we can have some really good discussion afterwards. Yeah, so basically everyone here, I'm sure, is familiar with the idea of local adaptation, and I like to try to use understanding local adaptation as getting a window into how evolution works. I hope that it can kind of give us some understanding of how the process of adaptation works more generally. But in order for it to do that, we need to understand how local adaptation differs from global adaptation and what's particular and special about it, and then use that knowledge to make light of empirical work we do on local adaptation. So I think one of the really big questions that I find fascinating is what constrains the diversity of adaptive responses. So if two species inhabit the same kind of broad climatic region or spatial region, they have various local adaptive problems. Will they solve it in the same way? Will they use the same genes, or will they find different ways of dealing with it with different genes or different phenotypic responses? And most of the work I look at is looking at the genetic stuff. And so asking how constrained adaptive evolution is. Is it flexible? Can you do a different thing each time? Or is it highly channeled, where really the only way to deal with a particular environment is to do this particular sequence of adaptation at different genes? And this involves lots of big problems. The first I'll think about here is, what does adaptation actually look like in the genome? And the second, how do we actually study what factors might constrain this and channel it one way or the other? So I'll talk today, a very brief review of some population genetic theory and some theory work that I've done that builds upon this, just to look at how this theory plays out in the genome scale. Then I'll talk about some empirical tests of theory, where looking at rearrangements in sticklebacks and some convergence studies in various plants. So just to start off, this may be quite familiar to many people in the audience. Two-patch models have been used to study local adaptation for a long time, where individuals migrate between patches at some rate, and natural selection pulls allele frequencies apart or eliminates the non-locally adapted individuals. And all the way back in 1930, Haldane and later Wright and others showed that a locally adapted allele will be lost when the rate of migration tends to exceed the strength of selection. So polymorphism won't be maintained in such a case. And if you think about different alleles with different effect sizes, this gives us this prediction that large alleles should be more likely to contribute to local adaptation over the long run, because they're able to resist this swamping effect of migration selection. The other thing we can think about is how migration selection balance plays out across a chromosome. So if you have a single mutation that's already present in the population, locally adapted, what happens when another mutation invades? And generally, theory has showed us that at high migration rates, there's more maladapted alleles present in the population at a higher frequency. And so new tightly linked mutations will be more fit than loosely linked mutations because there's less recombination. They'd like to be on a background with another good locally adapted allele, and that gives a general advantage for reduced recombination or tight linkage. And that can play out in various ways. So a general thing you could take from a lot of earlier theory before any of my work got to it is that migration selection balance favors what I've called concentrated architectures, so things that have fewer, larger, or more tightly linked alleles. And I think I'd suggest that a lot of these well-known examples of local adaptation by large effect alleles that I'm showing you here represent high fitness architectures. But the question is, did they actually evolve for that purpose? Lots of people have thought about the fact that these might just be the kind of shiny, glittery gold, to use Matt Rockman's term, worrying about whether we just find these things because they're big, and actually lots of local adaptation may be not typical to this kind of thing. So lots of questions surrounding this. Does nature actually ever optimize genetic architectures to look like this? How does global versus local adaptation differ in the underlying responses to these processes? And how does this inform our understanding of constraints to an adaptive response? So lots of my work has worked around this, and lots of other people as well. And I think these are really important questions to think about. And I'll try to kind of feed back to these as we chat. So the first thing I'll talk about, a really brief look through some theory on evolution of clustered architectures, in this case by rearrangement. So looking at how this advantage here that we're talking about, the advantage of linkage, plays out when you have a particular model. So the model I'll use throughout the theory work is a two-patch model. So we have two patches in a heterogeneous environment with migration, so migration between the two patches. Phenotypes are made by an additive contribution of the different alleles at the underlying genotype. So there's diploid individuals and mutations at many, many loci. Those mutations add up additively to give you a phenotype, and then that phenotype experiences selection relative to some optimum. So as the phenotype goes further away from the optimum, fitness is decreased, and there's a different optimum in each patch. So there's stabilizing selection within patches and divergent selection between them. And that results in there being epistasis for fitness when there's high genotypic redundancy, which I'll talk about later on. But basically there's additivity for phenotype, but the fitness function results in epistasis, which does some interesting things. Now, what we're looking at in these particular models is looking at the effect of transpositions or translocations, which can move loci from one place to another in the genome. And I'm not modeling a particular sequence of genetic cut sites and replacements, just generally just saying a rearrangement happens by cutting and pasting a locus from one place to another, or copy and pasting. And I've examined both in the paper that I'll talk about here, but just show you results from one of them. And really, the results are very simple. There's very striking visual patterns that emerge when you simulate evolution in either a spatially homogeneous environment or a spatially heterogeneous environment. So here you can see that the homogeneous environment, clearly each of the blue points is showing where there's an allele or a locus that's contributing to adaptation. They're separated by neutral loci in between them along this chromosome here. So from 0 to 50 centimorgans on the chromosome. And we're looking over time, and you can see there's really no change in the architecture for the homogeneous one, but the heterogeneous one evolves very rapid clustering. There's all sorts of interesting complications that come in here. I'm not going to go into them too much, really just specify this as a very broad and robust prediction that happens more or less quickly depending on the strength of selection, the population size, migration rate, the kinds of things you might expect. So summary of this first little bit is that highly clustered architectures can evolve via rearrangements when local adaptation is strongly favored. But I think this begs a really important empirical question, is whether real world biology will actually permit the evolution of clustering. So a really important consequence of rearrangements, of course, is that they can disrupt biological function. And it's not clear if when you move a gene from one place to another, it actually maintains any kind of integrity in terms of its expression landscape and the promoters and enhancers that are required to make it work, all that kind of stuff. Also, you can result in dosage imbalances where copy number may vary depending on the mechanism of rearrangement. So that can matter as well. On the other hand, though, we do see that there's been very rapid turnover of sex chromosomes, and this evidence suggests that genomes may be flexible enough to evolve clustering. But local adaptation probably results in much weaker selection than the kind of strong antagonistic selection that occurs with sex chromosomes. So I think it's a very open question whether this matters in nature. The next theory point I want to cover is a very different take on this, looking at the importance of genetic redundancy and how this can result in changes in the architecture. So to quickly review what I mean by genetic or genotypic redundancy here, this is when the number of loci affecting a trait is much larger than the number of mutations needed for an optimal phenotype. So here I'm saying that this 5 mutations in this little cartoon example can give you some optimal phenotype. The genome has 500 loci that affect redness. And so in this case, there's many, many more loci than needed. And so there's a lot of different ways you can make the same phenotype. And that redundancy is a property of the trait and the genotype to trait mapping that has some really important consequences. So I'll talk in this section about N optimal being the number of mutations needed to get you to the optimum versus N tot being the number of loci that can furnish those. And if you want to read more about the concept of redundancy, I was just working on a review with this recently with Aki Larsson and Katie Lauterhaus, so that we talked a little bit more about some of these concepts in there. But you can understand it kind of in terms of a landscape where many different genotypes yield the same phenotype. So again, we'll have 2 patches with migration, and what I'll show you are some results after 50,000 generations of divergence. So that they start at the same place, they diverge towards different optima, and then we'll look at them there. So here, this is the amount of divergence on the y-axis we see over time as a function of the per locus mutation rate underneath the trait. So each trait, you can kind of think of each locus as a QTL that has some mutation rate. And then the populations would be optimally adapted to their local environment when they have a divergence of 2 in this simulation. And so we can see that the highly redundant architectures evolve much, much more than the highly non-redundant architectures here. And the number of optimal mutations you need to get to the optimum is 50. Here, these ones are very highly redundant, and you can see there's much more happening there. I think what's really interesting, though, is to get at the underpinnings of this. And so we'll look at the underlying genetic architecture across different cases within this one kind of slice of parameter space. What I'll show you here is first the kind of conventional understanding of local adaptation from a population genetic perspective. We're dealing with swamping resistant mutations here. So when strength of selection is strong on an individual allele relative to migration. And I'll show you the phenotypic divergence here. So that what happens between populations, the phenotype, distribution of effects, and then a snapshot through time of the architecture. So here, this is when they're swamping resistant. We see lots of local adaptation evolving. They're just about at the optimum, which is 2 in this case. Distribution of effects per locus, we're seeing larger effects that are contributing in the direction of local adaptation. That's the red direction here. And then the snapshot through time, it kind of looks like what you kind of imagine local adaptation by high FST to look like. We've got stable, high allele frequency divergence between populations that lasts for, over 2,000 generations. When we go to swamping prone mutations, and this is when migration is strong relative to the individual selection coefficients on each mutation, we can still see there's phenotypic divergence, but there's many more mutations present and not as many large effect mutations. And we just see this transient architecture that looks more like static. There's no particular preponderance of blue over red. I mean, there is statistically, but you wouldn't see it jumping out at you by eye. And there's really nothing salient from one generation to the next. And this is interesting because if you took a snapshot of time at 48,000 generations and you characterize the genetic architecture, even if you could do it perfectly, it would be a totally different architecture than would be present at 50,000 generations. And so this suggests that adaptation by this mode would also be highly non-repeatable from one place to another. So if it's changing rapidly in time, it's also likely to change rapidly in space as long as migration is not really strong between different populations. An interesting effect that comes out when we're looking at really high mutation rates, and it's not clear that these kinds of mutation rates per QTL are ever actually realistic, but an interesting property that comes out is that you can go from, even when you're in an architecture that's swamping resistant, so individual selection coefficients in both of these cases here are strong. When we go from a low mutation rate to a high mutation rate, you transition from a stable architecture to a transient one. And that's just because at the high mutation rate, there's so many equally fit possibilities present that they end up drifting with respect to each other. And so there's not, even though selection could be strong on individual loci, this is a property of redundancy. When there's lots of different ways to make an optimal phenotype, they drift with respect to each other, and you tend to see lack of consistent architecture over time. And so I'll get into why that matters in a little bit as well. But what I found this is really interesting and informed my understanding of local adaptation, because it shows that swamping, this kind of population genetic limit, doesn't actually provide a hard limit to local adaptation. The response just becomes quantitative genetic. The amount of divergence depends upon the mutation rate and the genetic variance present in the population, but there's no underlying stable architecture. So swamping doesn't prevent stable architecture, swamping does prevent the stable architecture underlying divergence. So you won't see really high FST at the underlying loci, or you won't see big allele frequency differences that last a really long time when swamping is predicted. So population genetics is still very relevant for telling us what the individual alleles are going to do, but it turns out it doesn't give us a lot of insight into what's going to happen at the phenotype. And I think what I hope is clear from this is that this idea of redundancy has a very critical impact on adaptation. If you have low redundancy, not very many ways to make a given phenotype, you're going to see high repeatability, because many different populations in the same environment will probably evolve exactly the same way, because there's not many ways to get there. Whereas if you see high redundancy, we may see lower repeatability. And I think that'll really, a way to study redundancy that I'll get to later is to actually look at repeatability of adaptation and try to make some inferences of what this critical parameter looks like. So I think a very clear picture from a lot of this theory is that you can see very, very different architectures depending on the underlying genetics, either highly clustered or highly transient, even when local adaptation is happening. And I think the empirical question now is how commonly we see either of these two architectures in nature, why, and what that means about the underlying biology. So I'm now going to switch gears and talk about some work in my lab that's been working on empirical tests of some of this theory. So I'll start by looking at whether rearrangements build on genomic islands in Stickleback, and then talk about using convergence of local and underlying local adaptation as a way to study constraint and redundancy. So I'm going to fly through some of this a little bit quickly, and I apologize for not going into the methods more. I find talks that don't go into methods enough frustrating, and I'm committing that sin here. So please ask questions if you like specific details of the methods more in the question period. Basically, what motivated this study was the kind of visual observation that Stickleback, when these early genome studies in Stickleback were done, and there were multiple populations between freshwater and marine environments that had colonized freshwater environments in various different places around the world. They looked at the underlying architecture of that and found that often the same regions of the genome were involved in those colonizations. So here, this is this well-known locus Eta, and you can see the green dots and the height of these bars on the Manhattan plots are showing you where there's loci that seem to be contributing to divergence between marine and freshwater. And we see, big clusters on chromosome 4, cluster on chromosome 21 with an inversion, cluster on chromosome 7. And these are also chromosomes where other QTL analyses have shown a preponderance of QTL effects affecting various different phenotypic traits. So using various lines of evidence and talking with people that work on Stickleback more than I do, I kind of a priori decided that chromosomes 4, 7, and 21 show particular enrichment of FST and QTL outliers, and they show these kind of genomic islands, and we want to know whether or not rearrangements mattered for building that. So that's the kind of question we're trying to address here. To do this, I worked with Qiushi Li, who's a postdoc in my lab, to do a de novo assembly of the tube snout genome using PacBio and Hi-C. And the tube snout is this Olorhynchus flavidus here. It's kind of the first outgroup beyond the Stickleback clade broadly written. And then we also looked at Asian sea bass as another outgroup. So we did comparative studies with this new tube snout genome, 3-spine stickleback, Asian sea bass, and then also used a new Amur stickleback genome to help with some of the analysis. We wanted to first look at macro rearrangements, and some of this was known from previous work using linkage maps. They'd known that there were fusions in stickleback on chromosomes 4 and 7. We helped the tube snout genome helped show that chromosome 1 was a translocation in this case. And then we also identified a new kind of complex set of rearrangements at one end of chromosome 21. And so it was really interesting for me to notice that all 3 chromosomes most strongly implicated in local adaptation had also experienced macro rearrangements. And I think what's really fascinating is that chromosome 4 fusion had actually independently evolved in 9-spine stickleback. And that was known before our work, but we just kind of realized that's amazing that 2 fusions involving the same daughter chromosomes or grandparent chromosomes fused to make this fused daughter chromosome. So it's some interesting evidence. A lot of this is kind of a little anecdotal and hand-wavy, but that's how it deals when you look back at these retrospective studies. What I was most interested in was looking at the distribution of 3 kinds of small-scale changes in genome architecture. So looking at single or multi-gene rearrangements along chromosomes, just like the theory I was showing you, where single genes moved apparently between one chromosome and another. We did that based on a synteny analysis. Looking at tandem duplications, where there's been a duplication of the same gene in a chromosome. And then cases that might be representative of de novo gene birth, where there's lineage-specific genes present in stickleback, 3-spine, and Amur stickleback that are absent from every other species we can find. And what we wanted to do was ask if these were clustered around candidate genes known to contribute to local adaptation. So I'll show you some plots that look a bit like this, where we're looking at chromosome 4 here. And I've just shown you various lines of evidence here for regions of the genome that are thought to be important in local adaptation. So these are FST outliers between marine and freshwater, extreme divergence between marine and freshwater, multiple QTL, and lots of QTL number from a meta-analysis done by Peichel and Marquez. So we'll also show you on these chromosomes the ancestral fusion, if there was one. So here's the break point between the 2 halves of the chromosome. And then these candidate genes that are known to affect local adaptation in stickleback. And we're going to look and see if there's clustering of these rearrangements around these genes. So here's the results. We're showing you the 3 types of event here: rearrangements, lineage-specific genes, and tandem duplications for chromosome 4, 7, and 21 here. And here's our candidate genes in purple. There's the complex rearrangement that I mentioned on chromosome 21, and there's the fusion on chromosome 7. And what we did was test significance of whether there was clustering here. I mean, by eye, you can see some things that look like clustering, but is that really significant? So we would permute the location of all of these events within chromosomes, count up the number of events within X base pairs of a candidate gene, and then repeat this 10,000 times and test this across different intervals out to 3 megabase pairs. What we found was that there is significant enrichment around these particular kind of genomic islandy candidate genes. So we found enrichment of duplications around EDA and MSX2A, and then significant enrichment of all 3 types around this BMP6 TFAP2A region on chromosome 21. And interestingly, we found this was robust to spatial scale. So when I show you these as significant, they're significant at many different spatial scales as we tested out to the 3 megabase limit. We also looked at different density models. So we first tested under a uniform density model where events had a uniform probability across the chromosome. We also did this where we fit a density model to the observed number of rearrangements, or LSGs. That tended to peak near the telomeres. And so we would fit that to all of the non-rearranged chromosomes and then look at these particular ones as a model. And we found that this is robust. These results were robust to the choice of density model. So that's interesting. It's not a smoking gun, but it's kind of neat. We then wanted to see if there was some other data we could bring to bear on this. So Verta and Jones published a really nice look at differential gene expression between marine and freshwater genotypes. And so we took all the genes they identified as significantly differentially expressed and asked whether they were enriched in our 3 different types of a micro rearrangement event. So here you can see the rest of the genome. So these are the differentially expressed genes that are not rearranged, that are syntenic in all of the locations. And then these are the duplicates, and the rearrangements were significantly more enriched for genes that were differentially expressed between marine and freshwater. So over threefold enrichment here and over fourfold enrichment here for the duplicates. So lots more chance of being differentially expressed if a gene also went through one of these rearrangements or duplications. So again, it's nice, but it's not a smoking gun. So I was interested in this work just because it shows that there is, there's some signals that are consistent with what we would expect if local adaptation was indeed driving genome architecture evolution. It's not conclusive, I would say, and I'm hesitant to put too much stock in this, but at least does agree with what we would expect to see. So all the three chromosomes that were involved in local adaptation, often to marine freshwater, have undergone macro rearrangements, these fusions. We see enrichment on chromosomes 4 and 21 around some candidate genes, but not on chromosome 7, not around PIDX1. And we did see this significant differential expression, but again, none of the candidate genes that I was most, these ones, it would have been really neat if we'd seen that ETA had moved location or something like that. And that did not happen. So no smoking guns, but I found the significant differential expression result interesting. And we're doing some further work now on annotations with this work. So the last kind of broad area that I want to talk about is using convergence as a way to study genetic constraint. And I think that a lot of us are probably very well aware of examples of convergence. For example, MC1R being involved in local adaptation to color pattern differences and to different color environments in a whole range of vertebrates. And you kind of wonder, though, is that typical? Is that just a quirk of color pattern? Maybe color is really easy to evolve because all you got to do is knock out a gene or change something, and there's really just one way to do it. So you've got this really low redundancy architecture, and so you tend to see lots of convergence. And the question is this typical? And I think that's a really big and important question, and understanding why that varies is, I think, a really interesting one. So I want to put this back into the context of the theory I talked about before. Thinking about clustered versus transient architectures, we're expecting that convergence or repeatability of the genetic basis of local adaptation is going to occur more readily under oligogenic architectures, so smaller number of causal loci that can evolve into either clustered architectures or do that kind of thing we expect. If we see a highly clustered architecture, we might expect that that's more likely to evolve over and over again or be involved in local adaptation. If the underlying loci are themselves clustered, you've got this substrate that's already ready to go when you encounter some new environment. But then on the other hand, if we see most adaptation involving these alleles of small effect and small frequency changes, we're not going to expect any convergence. We're not going to expect any repeatability. And so we can kind of look at this both ways. We can ask, do the predictions that we see from local adaptation bear? And then also feed that back and say, based on what we see underlying local adaptation, what does that tell us about these constraints to the way that evolution actually works? So I think kind of big questions is, what is the repeatability of adaptation at the genome scale? How does this vary among traits at different levels of organization? You can imagine that very circumscribed simple traits, say like a secondary metabolite pathway, there's really not very many ways to make a terpene. And so things like that kind of pathway might be very, very low redundancy, whereas things like height growth in conifers or humans might be much more high redundancy. And so we may see different effects of trait level of organization, differences among species, differences in the type of adaptation, whether we see global versus local adaptation, and then always getting back to thinking about why, and what does that tell us about the process of adaptation and the way that organisms' body plans give rise to that. I'm only going to scratch the surface today. These are the questions I'd like to answer. This is kind of my big wish for our field. We're not getting very far yet. So some of you may have seen a paper we did a little while ago comparing lodgepole pine and interior spruce in Canada. We did a GWAS and GEA Environment Association analysis looking at 11,000 orthologs across 250 populations. And we found that there were 47 genes that showed strong signatures of local adaptation in both species. That was really neat. I was surprised and fascinated to see that we could find that. But the question that we kind of came away with is, okay, well, now what? What does that mean? And so, of course, more data is always the answer, or it's one answer. And so we thought we'd extend this to other species. And so working with Sally Aitken and others, we did this co-adaptree project to extend this analysis to several other species here: Western larch, jack pine, and Douglas fir. And this is a huge collaborative project. Many more people contributed than I've put on this slide, but I'd like to highlight in particular Pooja Singh, Mung Lu, and Brandon Lind, who are postdocs in our labs that have helped with all of this work, doing all the bioinformatics and the analysis. And I'd also just like to flag that this is highly preliminary analysis and highly subject to change. So this will be on YouTube, but if you watch this on YouTube later on, I'm gonna say, don't hold me to this. So the really fun thing that we got going with was Tegan McDonald, who's a master's student in my lab and is looking for a PhD eventually, did some fantastic work looking at making a linkage map in lodgepole pine. And this allowed us to actually make Manhattan plots in conifers, which if you work on conifers, that's a really fun concept, because usually we don't have much in the way of good genomic resources. So Tegan, we did sequence capture, and then she used the sequence capture data on tens of thousands of genes to make linkage maps in conifers. And that allowed us to kind of guess at, assuming that the underlying synteny was conserved between all these species, throw the orthologs onto those linkage maps and see what happens across the genome. So here we've got signatures of association to degree days below 0. So we're looking at many populations in each species and looking at genotype environment associations between alleles and environment, with degree days below 0, so cold temperatures being the environment, and then aggregating those per SNP scores using a method that I'm working on with led by really Tom Booker and Mike Whitlock, where to put together individual p-values into what we're calling a index of association, this weighted Z WISA index of association. And so here, the higher you are on the y-axis, the more evidence for that gene being involved in local adaptation or association. And you can see the position in centimorgans along the x-axis. I'm just showing you linkage group 1 here. And you've been squinting at this now as I've been blah-blahing, and I'm sure you've noticed some interesting patterns. Some that leapt out to my eye is that we see every single species, we see a large peak here at this linkage group 1, where Douglas fir coastal and Douglas fir interior are just two different, they're kind of subspecies of Douglas fir. So they are similar, they definitely share standing variation, but we're analyzing them separately for now. But really strong convergence, which I was surprised to see. You know, I really wasn't sure whether we'd see that, but also, and I never want to overemphasize convergence, because I think that non-convergence is biologically just as interesting as convergence. You know, we tend to focus on trying to find cases where there's this wow, kind of wow result. We can go and try and put it in some glossy journal. But I think that the complicated results are just as interesting. And so here, you know, we see a high peak in Western larch that's not found in any other ones. And we see a high peak in jack pine that's not found in any other ones. So there's idiosyncratic species-specific responses as well. And I'm just going to compress this in a really disgusting way. There's that, just to show you the lodgepole pine work. This is done in a different reference genome. So again, this is all a little hand wavy, but again, we see the same peak on linkage group 1 in lodgepole pine here, using different methods, but the same degree days below 0. We're seeing the same one contribute. Interestingly, jack pine and lodgepole pine, we're seeing also a shared peak there. But again, this is all a little bit hand wavy, but I think it's somewhat representative of what I kind of see when we stare at Manhattan plots over and over again for a really long time. You see some convergence and some highly repeatable patterns, and you see some idiosyncratic stuff and highly non-repeatable patterns. The real challenge, I think, and you know, we can talk about this ad nauseum, is trying to separate false positives from true positives. And I could go into that for an entire talk, so I won't talk too much about that here, but that's a really thorny issue. And identifying convergence is easy because the same false positive is unlikely to occur in many, many species. Non-convergence is a lot more thorny. Trying to see if this is a true positive or not for all sorts of reasons is difficult. So I think there's some really intriguing problems for us to solve as a field. And I think we're a really fun time to be looking at this data. Just to show you another line of evidence kind of like this and some other Manhattan plots we've been staring at lately. A recently published paper that I helped work on by Tedesco et al. Was looking at inversions in sunflower at various different species. Here you can see the sampling of all those different populations and the phylogeny of those species. And then we wanted to kind of take that data further and look a little more specifically at signatures of repeatability of adaptation across these species. So this is done with postdocs in mine and Lauren's lab, Shagag Sudi and Moshtaba Jahani. And again, this is collaborating with Lauren Riesberg and many people in his lab to look at this data. So I'll show you similar kinds of plots to what I just showed you before. Here we're looking at Hargreaves evapotranspiration index and 4 different species or subspecies, Annuus, Argyphilus, Phallax, and Pedularis. This is just looking at chromosome 13. Along the x-axis is the position. And here this is a different index of how strongly associated given 5 kb windows are. So if a 5 kb window has an enrichment for outliers related to this Hargreaves evapotranspiration index, it's got a high score there. And we can see some cases here where we see something that looks like convergence. So here in Argyphilus and Annuus, we see a very big peak in the same genic region here. But that's not shared with Phallax and Pedularis. Again, here we can see the same peak in Annuus and Argyphilus at this other region. It's somewhat shared if we just, these red dashes at the bottom here are what we've looked at clustering based on linkage disequilibrium. And regions within a given red bar tend to be in high LD with each other. So it's not clear whether you call this as a cluster or not. I mean, here we've got the same, another peak at a very different gene, but in high LD with this one here. And I think this just starts to get at some of the problems involved in trying to make inferences from Manhattan plots about what the architecture of evolution really looks like. Of course, lots and lots of signatures, just as before, are unique to one species. We see lots of peaks that are particular to one species or another and are not shared between species. And so I hope that kind of theme is clear to you that we do see significant convergence and more than you'd expect by chance, but we also see tons and tons of non-convergence. Here's just another way of looking at that. Here's the same index that I showed you before on the y-axis, the low to high here. Red is very, very high on that y-axis. Here is the trait or the environment with the highest repeatability, this EREF, the Hargreaves transpiration index. And here's the one with the lowest repeatability, leaf shape index. You can see there's really not, this is looking genome-wide, so not just at the chromosome I showed you. There are some cases where you see repeatability between particular genes in different windows in different species, but lots and lots of the strongest signatures are found only in one species or the other, and sometimes in some cases we see almost no overlap whatsoever. Again, parsing false positives from true positives is an ongoing challenge, but I think that what this shows us is it gives us this broad idea of there being some repeatability but lots of non-repeatability. And so I think that the challenge now, as we accumulate more of these data sets, is to think about, when we observe variation in architecture between different traits and different species, what does that all mean? How can we make some tests that get us to a more process-oriented or a more kind of cohesive understanding of why this is happening? Do these two different observed architectures just represent random draws from the same underlying process? Or is there something fundamentally different about adaptation to EREF versus variation underpinning this leaf shape index? And I think that question is a really important one. So to get at this and to evaluate what drives repeatability, we need to have some means to quantify it. And I've done some work that we'll talk about now is kind of thinking about how we can move towards this. And the way I've approached this problem is to compare the observed repeatability that we see to some null expectation under the idea that there's no constraint. So we kind of assume that all genes in the genome could potentially give rise to adaptation. And that's kind of what we do under, say, an infinitesimal model or a universal pleiotropy model, where every gene in the genome could potentially affect a trait. And then, you know, if you can reject that, see if there's some alternative model with a reduced effective number of genes that could fit better. So that's kind of the broad conceptual way that we want to deal with this. I won't go into this method, but it was a really fun project I worked on with Eliza, Kay, and Mike that we published a little while ago in PLOS Genetics there. And broadly, the broad logic of what we did was we just said, you know, if you see a whole bunch of genes, some of which are adapted and some of which are non-adapted, we can make some index which we call the C-score index, which tells us how much more overlap do we observe than we would expect by chance under our null model. And so a C-score of 0 in this case means that adaptation is working as if it's a random draw from the genome. Like our null hypothesis is true. No particular gene matters more than any other gene. Any gene can contribute. If we see a C-score greater than 0, that means that something is constraining adaptation to some subset of genes in the genome. I think if you told this to a molecular biologist that's been working on, you know, figuring out the gene for this or the gene for that, they'd scratch their head and wonder why you were even making this model. But I think that, you know, because I think a lot of people would just have this idea, well, of course there's genes for X, Y, Z, and only those ones are going to be contributing. But I think this is a very useful benchmark model, and we can have all sorts of discussion about why it is. So I'll just show you a context of how you could use this in an ideal world. There's all sorts of problems. But just going back to this first data set that we published on lodgepole pine and interior spruce, here looking at adaptation to mean coldest month temperature, so one of the environmental variables of how hot or cold the environment is, you know, we found 71 putatively locally adapted genes in pine, 139 in spruce, and 5 that overlapped in that particular variable between both of them. So we could plug those numbers into our method here, and out of a total number of genes that we were able to sample, we see these things, and that gives us a C-score of 4.58. We got a number. That's something. You can kind of also work backwards and figure out, well, this is the same thing as saying if instead of 11,400 genes, there were only 2,000 genes that could have been drawn from, that would give us a C-score of 0. So it's kind of like saying it's as if adaptation is working from this smaller pool of loci that are actually potentially involved in adaptation. So it's as if these 5 overlapping genes come from random draws from a smaller pool. And that's how the method works. That would be really nice if that's how easy it was. I think there's all sorts of caveats involved in this analysis, which you know, we could really talk for a long time about each of these. So our approach assumes that you accurately know which genes contribute to adaptation. So it assumes that that's known with confidence. And clearly, you know, the small effect loci, if this is a real effect locus, your power to detect it is going to be much smaller. Oh dear, sensitive mouse. Your power to detect it is going to be much smaller than your power to detect this one. So we've got detection limits working there. Structure correction, when everybody that has done a GEA analysis has thought about structure correction, hopefully ad nauseum, because it's really important. But that can cause all sorts of problems about which kinds of loci are false positives or false negatives. Also, this approach assumes that genes are independent. You know, when we start with this draw here, this approach says that, you know, this gene here is independent from this gene here. That's how the statistics that we worked on work. But we know that LD is rampant in genomes, especially when strong selection is working. And we think that clustering may be happening if local adaptation works. And so that could really complicate things because there's non-independence. Some nice work that we recently published with Tom Booker and Mike Whitlock also looked at recombination causing real problems for tests of local adaptation. And that can really cause non-independence as well. A practical problem is just that orthology reconstruction is not equal. And so missing orthologs is going to cause you to have problems when you try to implement these methods. And also there's a growing awareness of copy number variation being incredibly important for local adaptation or for adaptation in general. And so duplications per se may be involved in adaptation, and so you have to reckon with how to deal with a duplication that's present in one species but not present in the other. And we found evidence just incidentally in the lodgepole pine interior spruce work we did. We found that genes that were duplicated in one species or the other were more likely to be involved in local adaptation than genes that hadn't been. So that was an interesting little side analysis. And all sorts of neat work is coming out in copy number variation, showing that copy number is a way to really rapidly change things like gene expression, flux through metabolic pathways, like secondary metabolites especially. So we may see that really mattering, and accounting for that is critically important. So I think that our method was a really nice, ideal way to do this. I think there's so much room for improved methods, and I'd love our field as a whole to be able to come up with better ways to do this. We're continuing to work on this, and I'm really interested in that in general. So that's broadly what I wanted to talk about today. I think what this hopefully shows you is there's so many open questions in this field and so many things to look at. You know, how commonly we see different things, concentrated versus diffuse architectures underlying local adaptation. Does that differ between local adaptation and global adaptation? Global adaptation, you don't have this advantage for clustering or large effect alleles, and so seeing whether that changes. Do we see stable or transient architectures in different kind of contexts? And what's the repeatability of adaptation? And I think in all of these, you know, establishing how commonly these occur is the first step and a really important step, but it leads then to the question of, you know, why? Why do we see one thing or another? What is the contribution of the different evolutionary processes driving the answers that we're seeing and the variation in those answers? And hopefully we can get to some idea of what factors constrain the diversity of adaptive responses. Kind of two really caricature ways of thinking about this that I think I'd love to learn in my lifetime is, you know, how commonly do we see evolution being just, you know, there's only a limited number of phenotypes that can be generated. You know, there's just not many ways to make a given phenotype with your underlying genome. That's one world. Or we may be living in a world where there's lots of ways to make a phenotype. There's high redundancy, but if we see repeatability, it's because not many of them are very good. Either there's pleiotropic consequences, or you need a clustered architecture under migration selection, or various other reasons. And so I think that understanding whether we're in kind of a world that's characterized by one kind of constraint or another is just a really nice, broad idea about how evolution works that I'd love to understand better. If you're interested in this, there's a postdoc opportunity in my lab that's recently come up that's really, I think, a remarkable chance to look at these questions. Clément Rougeux has been working with me for the past little while, and he's done some incredible legwork. Clément's moving on back home to France to work on his family business. An opening came up in his family business, and he's decided to move on to look at to work on wine and foie gras. So I mean, I know it's an amazing opportunity, and I'm sad to not be working with Clément. He'll keep on working on this project, but he's done some amazing work. We've got over 40 collaborators and 51 species, and Clément has recalled the VCFs on a common pipeline so that we can compare all these different species looking at local adaptation. The data sets are huge. We really picked the ones that had genome scale data or exome capture data. So we've got about 1.5 million SNPs an average per species. And you can see here, they're representing a really wide range of environments. And Clément, as I said, has done an incredible job of collaborating and getting everybody to submit data and get this up to a point where we can analyze it. So if you'd like to work on this with me, please do get in touch. This can be done remotely, given the way that the world works these days. So please contact me for details if you're interested. With that, I'd love to thank everybody that I mentioned specifically here, and many more people more broadly that contributed to this work, and funding agencies as usual. And thank you very much for all attending. I really hope we can have some great discussion about this. So yeah, that's it.
Thank you so much, Sam, for this excellent presentation. Thank you so much.
stop sharing maybe, or so we can see the room, or what's the best way?
Yeah, you can stop sharing, and in case we need to go back to your slides, then you can start sharing again. But for now, I think it's all right.
Sounds good.
Yeah, so now we've got some time left for the questions indeed. And the way you can pose the questions is by raising your hand, and the raise hand button, you can find it under the list of participants, which you can find on the lower bottom bar of your Zoom application. So you click on participants, and then under the list there is a button called raise hand. And you can also use the chat for your comments and also for posing questions. So feel free to use those modalities. Just in case you are not seeing it, Sam, thank you for this inspiring presentation from Fabienne Laroche. Great talk, Ivan Scotti. Okay, we've got some questions that are coming up. And the first one is from Tanya. So if you can also just please introduce yourself, where you're coming from, that would be great. Thanks.
Hi, Sam, again. So I'm Tanya Pyhäjärvi from University of Oulu, and I wanted to ask about this, the role of genome architecture in terms of recombination, or like, let's say, where the transposable elements, or like, how do you think do they affect, like, let's say we have a lot of heterochromatin somewhere with the reduced recombination or repeat elements that are the breakpoints. So do you think they could actually have important effect on what goes on?
Very much. And actually, I cut a slide that would speak a little bit to that. So maybe I'll just, if you don't mind, I'll share one slide that I could have showed you. And then I'll talk more broadly about that. Everybody can see that? So this is here, the transposable elements in Stickleback. So you recall, these are the candidate genes I showed you. And you can see that the TEs tend to peak around the ancestral breakpoints in both Stickleback and tube snout. That persists around these breakpoints. But what we found really interestingly, and that was significantly enriched, around this region, this is the BMP6 TFAP2A region that had significant enrichment of duplications, LSGs, and rearrangements. There was by far the biggest enrichment of any TE anywhere in the genome was found just upstream of that. You can see here the difference between the red line in tube snout and the black line in Stickleback shows you just how crazy this area is. This is this LTR ERV1 element. If we express it in terms of relative enrichment, you can see those two windows that make up the part in there are just way, way, way outliers, by far the strongest enrichment of any region in the genome. So I think that your question is totally on point. I think transposable elements are basically the way genome architecture evolves. Often, because they can facilitate presumably things like ectopic recombination. Certain elements can actually do rearrangements themselves, like the helitrons and other kinds of ones. So yeah, I think transposable elements are fascinating, and I think huge questions about what our genomes would look like if there were no TEs, and what adaptation would look like if there were no TEs. Yeah, I think that's an area for so much fruitful research. Maybe I just blabbed there. Did I answer your question?
Yeah. I was just thinking that because there are so much of them in conifers, and maybe mostly like to ignore them. It's just like maybe we should pay more attention.
I wonder. Yeah, certainly the dogma within conifer genomics is that there's been very few rearrangements based on some comparisons of linkage maps. So it seems like synteny is quite conserved. And we did. Tegan McDonald, I mentioned, had done looking at this linkage map she did. We tried to find if there were any evidence of things that were anomalous and looked like clustering or looked like rearrangements. And in no case did we find any that looked like rearrangements. So we're still wondering if that happens ever in conifers. I'd be fascinated to see a more dedicated study of that. As you all know that work on conifers, with our genomic resources, it's challenging. But I think those resources are improving dramatically. every year. And so hopefully we can get at that better. I think I'd love to see more work on that.
Okay, we've got some hands coming up here. So the next one is Felix. Felix, please go ahead.
Hi, Sam.
Hi.
Thanks for your talk. My name is Felix Kugerle. I'm from WSL Birmensdorf, where we visited a couple of years ago. So actually, my question went into the same direction as Tanya's, because we looked at TEs in the Brassicaceae and found a tremendous rearrangement and a lot of TEs all across the genome. So in addition to what Tanya was asking or you were explaining, do you think that the effects of the TEs mostly occur in or by rearranging the genome or by moving genes along, or would it be rather by jumping into promoter regions or into exon regions and by that promoting or silencing these genes? Do you have any guess here?
I wouldn't hazard a really strong guess. I certainly wouldn't put money on it. I would guess that most, by frequency, most rearrangements, maybe their biggest impact is just on facilitating ectopic recombination or those kinds of things. Whether or not a TE really moves a gene and it becomes useful, I think that's another question, because I can imagine so much gets disrupted if a TE happens to actively move a gene. But if it does, that could be really fortuitous and a very kind of low stakes way of getting a big rearrangement. Because basically the more of the genome that you're rearranging at once, the more chance you're going to cause gene balance, like dosage balance problems and stuff like that. So I don't have much based on any empirical data. I remember some interesting work done by Woodhouse and Freeling on Arabidopsis suggesting that TE-mediated gene movement was rampant between Arabidopsis and papaya, but you know, I haven't seen a lot of follow-up work further. So I'd be very curious to see empirical data that would inform that more.
Okay.
I'd say it's an open question from my perspective.
Okay, thanks.
Thank you.
Thanks, Sam. We've got some more questions coming up. Moritz Ehrlich. Moritz, please go ahead.
Hi, Sam. First of all, great talk. Super clear explanations on a very, very complicated subject. To introduce myself, I'm a PhD student at the University of Miami with Douglas Crawford and Margaret Lexiak, and I actually work on extremely short-term local adaptation at a very small scale. So I actually look at within generation selection to these local niche habitats. So I'm actually very interested in your idea of transient architectures, especially in the context of where we take migration to the most extreme. So where we have a migration rate of 1 and we actually see, you know, panmixia among populations every generation. Because I think this sort of architecture would allow for some probably small divergence to persist through time, but with transient architectures every generation that could actually maintain genetic variance. So I was wondering what your thoughts were on that, and whether we can actually, or whether we could expect local adaptation at much smaller scales. We just don't see it.
Yeah, I mean, I think that in those cases, I think it makes sense that you could call that local adaptation. You know, I think you get a bit of a fuzzy boundary when you've got such high panmixia that, you know, in the classic models at any given, you know, it kind of depends on what your frame of reference is. From the perspective of a given allele or a given gene, it really kind of wants to be non-polymorphic and non-locally adapted so that it's kind of good everywhere. But that's not the way, you know, selection works on phenotypes. So I think that, you know, I think that that's really fascinating to look at what happens in those cases. Whether or not it maintains more variance or not, I'd be curious to see data on that. And yeah, I mean, I think it makes tons of sense that, you know, you can see, you know, a response to selection is proportional to your standing genetic variance, and that response to selection could provide some kind of durable phenotypic divergence, even with very, very high migration, or maybe not durable. Yeah, I think it's a really neat area to look into and worth looking at. I think very challenging to get meaningful stuff on the genomics out of that. And so it's, if you can see a signature that's at the genome level, that would be surprising and interesting.
Thank you.
Thank you, Sam. I just wanted to say that normally when we have physical meetings and somebody wants to comment directly on what has been said rather than wait in a long queue to ask questions, I would tell them to raise both hands. And in that case, I would know that it's I don't have to go in a chronological order. Since we don't have that option on Zoom, I suggest that anyone who wants to comment directly rather than raising hands, they can also just put something in the chat. So then I will know how to prioritize the comment or question. So we can also have some kind of discussion rather than just taking the questions one by one. If anyone wants to comment on that, please feel free. But anyway, the next one is Santiago.
Yeah, actually, I have many questions, but I'm going to make one, and then we will see how it goes. Like, and I wanted to make also a comment. I think it's actually what Tanya, associated with Tanya, was saying before. It's true in the forestry community, we have been ignoring anything that was just not mutations. So, and I think I don't really know why. I mean, must be historical reasons, I guess. Like as you say, because of these early thoughts that genetic maps were very synthetic or this comparative mapping, and it's a little bit worrying that we may have been missing the main story of adaptation there. So like, so it's a really nice insight for our community to. It's clearly a direction we should work more. But that was just a comment. I wanted to make a question. Like you said something that is actually is also worrying me a bit. Like because very often, like we don't have many good candidate genes in forestries. We don't have the capacity to do the kind of experiments people do in Rapidosis or in Drosophila or like. But then we are always taking like if you get the same candidate gene across different species, it's like a proof of that this gene is really important. I'm thinking, for instance, the hadrins, like it's a popular gene for tolerance to drought that has been studied in several trees. But now something you say in your talk, and it's also so myself that maybe also we can find the same false positives across species once and again. And I wonder if you have any ideas how this can happen, if it's a question of particular regions of the genome that may have different mutation rates, and this is confused with signature selection, or well, what is your opinion of that? How probable is that we are really just looking to these false positives in different species, and why?
Yeah, I mean, I think the chance, the chance of the same false positive in different species becomes small if you see the same thing over and over again, right? That under any null model, that if your alpha is 0.05, that just gets progressively smaller and smaller the more times you see it. So I think that if I understand your question correctly, I think that's rare. But if we just fail to see something because biologically the substrate for it is not there, that may be. Things like recombination rate do affect the propensity of, say, small mutations to be able to get over the migration load, the swamping load limit. That kind of thing might drive differences between high recombination rate and low recombination rate regions of the genome. I forgot to mention with Tanya's work also, seeing work in her lab with Jaco has shown that there's an inversion in Sylvestris, Pinus sylvestris, that may be really important for adaptation. So I mean, seeing that underappreciated side of conifer genomics, whether these kind of things that have been very hard to study might really drive some of the adaptive processes. That's a really interesting question, I think. So I mean, it's a vague answer, I guess, but I think that's one level of reason. Yeah. Another possible level of reason is, I mean, there's just, candidate genes that come from angiosperms. There's been a lot happening since the angiosperm-gymnosperm divergence. And so that there may be biological reasons why just the pathway that was used in angiosperms is not used in the same way in gymnosperms. And I think that's an equally interesting question.
Yeah. Okay. Thank you.
Okay, we've got Jobran Habib. Jobran, please go ahead.
Hi, Sam. Can you hear me?
Yep. Hi, John.
I'm a postdoctoral researcher at the University of Edinburgh. I'm interested in evolution of genetic architecture, which is why I'm here listening to your talk. My question is about maybe, and might be a naive question, is how do you distinguish multiple loci that are affecting the same trait or fitness or phenotype that are clustering versus a selective sweep? Maybe, obviously, you probably thought about this before, but I just have no idea.
Yeah, I share your concern with that. I think it's a very tough thing to get at. I suspect that the way forward with that may be through getting back to QTL studies. You know, if you can find especially a cluster that happens over a pretty wide region of the genome, if you take individuals from the extremes of a range and do a QTL cross on those individuals, if you do multiple QTL crosses, you might be able to decouple causal variants that are close together, but not so close that it's impossible. And there have been some really neat studies done back in the day where people were kind of curious about whether a given large effect locus that seemed to have a genetic correlation was due to pleiotropy or linkage. And you can look back at QTL studies that were able to resolve that and show that it was due to linkage. So I think that often with, for someone like me that spends a lot of time just doing genome scans and observational data, I think experimental work where you do crosses is going to shed a different kind of light on those kinds of questions. If you look at the Stickleback work, for example, we can see very, very wide genomic islands that have this kind of signature of lots and lots of extreme outliers over a very wide region. And that's because with strong selection, you can entrain a pretty large region of the genome in high LD and get that benefit of clustering, which is high relative to recombination rate. So those kind of very wide clusters are probably the ones to target for QTL studies, because the LD is being maintained by selection, not by actual physical linkage. So I think those kind of, I think that might be a best way to get at it. You can do some ad hoc stuff just with genome scans and just see, do the peaks do two peaks go down and back up again? And that maybe seems unlikely to happen, but I can make arguments that could be seen under particular forms of linkage disequilibrium and selection also. So I think that getting at causality in clusters is a really tough but important problem, and I would challenge the experimentalists that are smarter than me on that to come up with better ways of doing it. So that there's a dodge for you.
Right. Thanks.
Thanks, Sam. We've got Hernan next.
Yes. Hello, Sam. My name is Hernan. I'm a PhD student at SLU in Uppsala, and we've emailed a little bit about one of my projects. But I want to ask you now about adaptive traits that are, let's say, very simple in their architecture, being more, let's say, prone to convergent adaptation. And if you thought about examples in trees where we know there are major loci controlling these traits, such as the flowering time locus, that is very important for adaptation to phenology traits. And from the top of my head, I cannot remember if this can be considered a case of convergent adaptation, or if this is really the same ortholog in all the species that has been conserved because it's so important. But if you could use those as flagships to maybe really understand whether it is easier to find convergent adaptation on these kind of loci.
Great question. So I'll just clarify one thing I should have maybe explained more clearly. When I was saying convergent adaptation, I was really meaning convergent local adaptation. So not even that could occur from exactly the same homologous gene. The same gene is shared in two species, but as long as the alleles contributing to the local adaptation within each species were independently evolved, I've been calling that convergence. So it not requiring necessarily there to be a new gene evolving or something for it to be convergent. So it's a bit different than the usual use of convergent. It's a convergent polymorphisms using the same gene. But that's beside the point. I think, flowering time, we do see that particular one coming up over and over again. I suspect some of the things that we'll see most interestingly are secondary metabolism associated with defense, where you've got a very circumscribed pathway, like say like how you make terpenes, or some work in my lab. I've been collaborating with someone on working on opium poppy and how the poppy makes all sorts of different morphine and codeine and all these other compounds that probably had their origins in plant defense. There, you've got these very circumscribed pathways. And the nice thing is sometimes they're quite well known, and we could characterize, it would be interesting to do that and take a pathway where this is important. Let's go out and see what it does in nature in terms of variation in that pathway. I think that's a really interesting way forwards. I haven't seen a lot of work done on that, and I'm sure it's out there now, but I probably just haven't done enough reading. So I think that's a really neat way forward, is to kind of take, instead of this top-down genome scan approach, start from a set of known pathways and say, this is a pathway we know works. Do we actually see adaptation involving the phenotype that this pathway controls outside of that pathway? Right? Are there, to use the language of the omnigenic model, is you got your core genes and your peripheral genes. Are other things peripheral to that pathway important? I think that's a really interesting angle, and I think it's one that I'd love to see, and we're kind of having plans in my lab to do that eventually, kind of a candidate pathway inspired approach to these scans. I think that's really neat. Unfortunately, I don't, again, dodging for lack of time to read all the cool papers that are probably already out there, and just not sure if we know yet, but I think that's a really neat question.
So we've got Russell and Ben, but because Thomas has a related comment, I would like to give word to Thomas.
Thanks, and sorry for jumping in the queue. So Thomas Bataillon, I'm at the Aarhus University in Denmark. I had a related question to what was asked and what you commented on at the beginning of your talk, which is, so there is that sort of rampant problem maybe we all share, which is that most of the examples where genetics of adaptation and even more local adaptation have been dissected, we usually have at least one loci or one locus that has a very strong phenotypic effect. And I guess the emblematic example would be the EDA locus in sticklebacks, but there are many others. And of course, there is a related question for me, which is, do you have the impression that some of the measures that you propose, among others, the ones of redundancy that you didn't discuss so much, but at the beginning, are they all kind of assuming that you have actually a number of associations or QTLs that explain a sizable amount of the standing genetic variance, and that you're going to be powerless if you don't do that assumption?
I mean, yeah, any of those methods do require that you know what matters. And so we really are hooked when if there's lots of small effect alleles that really matter, that's a really big problem. And so I think that one interesting way to do this would be to start to, if you could get at proportioning, what's the percent variance explained in the phenotype or the environmental adaptation by the large effect alleles that we can study redundancy on, then maybe we can infer something about the rest of it by what's missing, kind of the so-called missing heritability problem applied to local adaptation, and inferring that, well, if it's missing, it's got to be low enough that it's probably redundant. But that's not a super satisfying answer. So I mean, it's a long way to go from the theory to the experiment and the inference. I mean, I think that our answers are inevitably going to be biased by the fact that the things that are non-redundant are going to be the easiest to find, and so we'll think nature is less redundant than it really is, just because of that. Again, full of non-satisfying answers today.
But maybe also makes sense in the sense that the concept you're trying to put forward about segregating redundancy that you discussed in the beginning, maybe they actually make sense for me, at least, to bridge between a single locus adaptation and a completely polygenic adaptation. Mm-hmm. So maybe there is also biological case to be made that there are a number of traits that are what I would call oligogenic. And of course, one can haggle about oligogenic, but maybe 5 to 10 loci that explain the bulk of the genetic variance.
Yeah.
And there are many traits that may be important. And you were citing repeatedly sort of monoterpene, and I have also an interest in that, but that maybe some of those traits of relatively low complexity might still be important, especially for those pathways. And that there is kind of hope to make progress without having the sample size of maize or humans or Drosophila.
Yeah. And I think that's, I've talked about, I think that gets at a really important point. It may be that, I've been talking about one trait as if the trait has a high redundancy or a low redundancy, but a given trait probably is characterized by portions of it that are non-redundant. And, the only way you can change that trait in that one way is to change the flowering time gene. But then, that gets you 50% of the way. And then there's a whole bunch of other ways you can get the rest of the way. So there may be kind of portions of variants that are non-redundant and other portions of variants that are redundant. And I think that's a nuance that I haven't built into any of this machinery yet. But I think it's really probably more the way biology really works.
Thank you, Sam. We've got 2 more questions, Russell and then Ben. So Russell, please go ahead.
Hi, Sam. I'm a student at the University of Calgary in Sam Yeaman's lab. I just had half a question about the clustering with the stickleback. I don't know if this is just because I don't know the history of stickleback, but so you found all these outliers that are clustered around the rearrangement sites. And so the story is selection is selecting for these rearrangements because it brings together these outliers, and a more clustered architecture is more fit than a diffuse architecture. How do you know that that's the story and that it's not the case that these big rearrangements occurred first, and then that created a barrier between the rearranged versus the non-rearranged, and then these locally adapted loci arose on that rearrangement background?
Yeah, that's a great, that's a really good point, Russ, and we don't know that. And certainly there's no way to get at the order of events in this particular case. I think all you can conclude from the data that I showed is that it's unlikely that the enrichment that we see around these candidate regions would have occurred by drift. That's basically all we can say. Is if you're in a world where rearrangements are just happening by chance all around the genome, they wouldn't have likely resulted in the patterns we saw. The order of events that might have led to this and all of that, it's unfortunately for now lost to time. And I don't know that we'll ever be able to get at that in looking at the stickleback themselves.
Okay.
Just because the signatures of those events are just long gone. That's a really good question. I think that shows the subtlety of some of these methods. You can make theory models that suggest that one or the other is more likely, but it doesn't provide the kind of proof that's particularly satisfying. Great question, Russ.
We've got a question from Ben.
Hi, Sam. Thanks for your time. I have a technical question. Sorry, VSL from Switzerland. Yeah, I have a technical question about the co-adaptree project. Really interesting. How did you compare the genome scans on multiple species? I mean, how do you deal with these very divergent tree species?
Yeah, well, so
yeah,
that's a great question. And yeah, I apologize for glossing over the methods. It's annoying when that happens. What we did was within each species, we used the closest species that had a reference genome if that species didn't have its own reference genome, and mapped the reads to that species. We then, Tegan McDonald had made this linkage map in lodgepole pine, which that had anchored all of the contigs for the lodgepole pine reference genome. And then Pooja Singh was working on ortholog reconstruction. And so she identified gene by gene, within each species, the genes that are orthologous to the lodgepole pine genome, and then mapped them onto the lodgepole pine linkage map based on the best fitting ortholog. And so that was how I made those Manhattan plots. I asked Pooja to do that specifically for this talk a few weeks ago because we wanted to rush that and just be able to look at that result. So that's, as I mentioned, it's really, really in progress, and we're actually revising that analysis and doing the orthologs a little better still. But the basic approach is that we have the linkage map provides the scaffolding of the lodgepole pine reference genome. We use orthologs to tie different the other tree genomes to that linkage map, and then show them on the show where they are along that linkage map. And so this excludes anything that didn't have a good ortholog. I think Pooja excluded anything that was duplicated in one species or the other, or where the orthology reconstruction couldn't resolve it. So we excluded a whole bunch of stuff from this plot that still we're trying to get better with the ortholog reconstruction. So it's a thorny problem, and anyone that's done orthology reconstruction knows how thorny it is. Does that answer your question? Any more nuanced questions?
Thank you. Yeah, I have another question if I have time.
Yes, please go ahead, Ben.
Okay, very unspecific but very interesting. What is the role? What do you expect? What is the role of environment on genome architecture? I mean, if we have a species with highly heterogeneous habitat, for example, would you expect a different genome architecture that compared to another species that is living in the very stable habitat? What is your expectation for these kind of things?
So when you say stable, you mean homogeneous in space or stable in time?
Good question. Rather in time. For example, if you have a high temperature range in some locations compared to others where it's very stable, you have a very low range, for example.
Yes, I mean, the theory, at least, all the theory I talked about today assumes temporal homogeneity. So there's no changes in the environment over time. And so if you have a heterogeneous environment within a high migration rate area, the higher the migration rate, the more that does select for a clustered architecture. And that emerges out of many, many different models. Basically, as long as local adaptation is beneficial at the phenotypic level, architectures that are increasingly clustered can be selected and favored to outcompete architectures that are less clustered. And that can happen either due to rearrangements or due to competition between alleles that are tightly versus loosely linked. And that would be expected to play out in the heterogeneous environment, but not in the spatially homogeneous environment. So you would expect that from the models. The question I think that's interesting is just whether or not real genomes work in such a way that that model prediction can come true. So it is a very clear prediction from all the theory, but I think the empirical question is then the follow-up. It looks like Santi has a follow-up question to that one.
Santi, is it a follow-up question? Because otherwise we've got two other, yeah, okay, go ahead.
Yeah, I was just going to say, what about like history traits? Also like condition of affecting genetic architecture, like longevity, for instance, or dispersal traits.
I mean, from the perspective of the models, any trait that you can represent, if the trait can be represented as being under something like stabilizing selection that's divergent between populations, then this architecture evolution will happen. An important point is if there's no redundancy whatsoever, if you need every single locus to give you the optimal trait, there's not a lot of scope for architecture evolution. So a lot of the architecture of evolution stuff I showed you is a particular consequence of a model that has redundancy built into it.
Okay, thanks, Sam. We've got another question from Chadley.
Hello, Sam. I hope you can hear me well. I'm Shadi Kastali from the University of Oulu, working with Tanya Pohayarvi. So you mentioned false positives, and I wondered if you considered the category in false positives where you have genes that are loci that are detected as locally adapted because you have for instance, an allele frequency very different in two different habitats, but it's actually genes that are haven't had the time to reach the population that is in the different habitats. So in a way, they are just temporarily, locally adapted, but eventually they will become fixed in the entire species. And so if you have thought of these, like, do you think they could be a big part of these problematic false positives or not?
Yeah, so I mean, I think that I'd call that a different flavor of false positive, because I mean, that's still adaptation. That's just global adaptation, right? It's adaptation towards a global optimum. And we actually just did a paper on this that's just been accepted recently, I think, to Evolution Letters, I think, with Tom Booker and Mike Whitlock. It's on bioRxiv anyway. But looking at you know, how often we expect global adaptation to be happening, and looking like local adaptation, just the way you suggest. You know, if you catch a sweep halfway through, it looks like it might be locally adapted. I think that that's important, especially in cases where you've got a very simple sampling regime, right? So if you're just sampling along a single cline, and that's the whole species is also existing along a cline, there's a much greater chance that some globally beneficial mutation is just midway through a sweep. If you've got clines going, you know, this way and that way and spatially crossing different environments in a very rough gradient, say like a mountainous environment where there's many, many mountains and you've got transects along each one, the chance of capturing a globally spreading sweep in such a way that it still co-varies with your environment becomes pretty small. So I think that if your species exists in an environment where you can use sampling design to prevent that being a source of false positives, or, you know, of getting the wrong kind of adaptation in your scan, then that's obviously something you should shoot for. Lots of species don't look like that. You know, a lot of times they're in a small enough range that you can't sample along a complex enough design to preclude that problem. So I think it's definitely there. The question of how commonly it matters is a big one. You can read our paper if you want to go more into that. And I think probably other stuff looking at that as well. Thank you.
Thanks, Sam. We'll get the last question from Pablo.
I think you may be muted, Pablo, if you're talking.
Yes, Pablo, you're still muted. We can see you now, but you're still muted. Please unmute your mic. No. This doesn't work. This doesn't work for us, Pablo. We can. This sound is really bad. It's maybe better if you just type your question.
That was a great question. I have no answer. I'm so sorry.
Pablo, if you want, you can type your question and I will read it. Unfortunately, this sound was not good. Okay, I'm not sure if Pablo is reading it. Unfortunately, I think we will need to skip it. But Pablo, if you want, we can pass on your question to Sam later on.
Please email me. That's oh, he's typing. Okay.
Oh, typing. Okay. So we wait.
Well, I'd just like to thank everybody that asked questions. That was really fun to have a great discussion. I really appreciate your interest and your participation, especially in Zoom, where it's sometimes kind of weird to participate. So thank you all for joining.
A silly one, just thinking about false positives and genomics island. And QLs.
QTLs. So yeah, any chance that QTLs are false positives by linkage to a large effect gene? I mean, I don't know if, I guess it depends what you mean by false positive there. I think if you do a QTL map, you know that there's some causal variant. If you find a QTL peak, you found, there's some causal variant there. The question is just, is that one causal variant or multiple clustered ones? So it's not, the issue of false positive then is like, are you, how do you kind of parse that further? So, if you do a bigger and bigger QTL experiment and eventually you find that you break up that one QTL into two separate ones, your QTL design is showing that those two separate ones are both affecting the trait of interest. So I don't think that's a false positive in that case. There you've shown that there's two causal loci that are close together that each have a detectable signature in your QTL effect. So if that's what you mean, I think that that's a QTL designed to follow up and try and disentangle what's going on in a genomic island. I think that that's not really like that. That gets at the problem of false positives. A given genomic island could just be one causal locus and lots of neutral stuff in linkage, and that's what I think this design that I was suggesting would be trying to get at. So I hope that answers your question, Pablo. Thank you. Please email me if not.
Thank you, Sam. I think it's time for us to close this because we've already gone a little bit over time. I would like to thank you so much, really, for joining us today, really dedicating the time. It's been a real pleasure. And we hope that EVOLTREE can indeed become such a platform for discussion on these big topics and linking different scientists from around the globe. So we are really glad to have seen such a large online audience here with us today and such a great interest in the topic. So thank you very much again to you and to the online audience. And I would like to invite you and encourage you to join for the last episode of this series on polygenic adaptation, which is going to take place next week, same time, with Neda Bargi. So with this, I would like to close it and thank you very much again and see you next week.
Thank you very much, all.
Thank you.
Thanks.
Thank you. Ciao.
Bye, thanks.
Thanks.